Now I'm going to switch and talk about the bitter rot, which seems to be an emerging and rising problem, especially south of Albany, and in some pockets where in Western New York, where the Lake Ontario is, and this is an issue which is quite different from black and white rot because it's caused by a different pathogen, Colletotrichum genus, and in there you have multiple species that cause this disease. And so far we have been operating in the Hudson Valley under the opinion that most likely the species that is dominant causing bitter rot is Colletotrichum furini, although we did not have any evidence so far. We now have that evidence, and most likely that's the dominant species, but we did not really, we just don't know whether there's more species because we do see a difference in symptoms. There's slight variations, as you can see here, not only from the early infections, but also there's difference probably because there's multiple pathogens causing this disease. But the typical telltale sign is if you cut the fruit, and then you can see this typical V-shaped wedge necrosis that you can see on the right lower image that I've presented. And also when you look at the this rot from the side, it's flat as a flat saucer, which is basically indicating you that it's most likely bitter rot and not a black or white rot in case. And sometimes, of course, when there is wet weather, when there's rain, these concentric rings that you see in the middle of the rot spot where it started, you can see these orange small droplets that actually are huge amounts of spores of the pathogen, asexual spores that allow the pathogen to be rain splashed and dispersed to new fruit that can be infected. So it's a growing issue in New York because we had outbreaks in 2015 and 16, and mostly it's around in the Hudson Valley region, but also people in Canada, in the Ontario region have issues with it, but also in Pennsylvania and other southern states like Kentucky and North Carolina and South Carolina and Virginia. So this is not an isolated issue that is just happening in New York. It's definitely widespread on the Eastern Seaboard. It seems that that many growers leave out their fungicide spray sometime during October with the late varieties, and closer to harvest there's no coverage by fungicides, and that's the way how this fungus moves in and then is being carried into the storage. It seems that the stem end of the fruit is the place where water keeps after the rain, and that's where this pathogen can infect, but the symptoms don't show up until you move the fruit into the pre-storage area. And then, while the bins are waiting to be placed in a cooler, they can start progressing. And then you put the bins inside, and then once you start packing, you see losses up to 5 to 7% in storage that we have seen in 2016 from bitter rot. So, and we have also seen in the field losses up to 60% on Honeycrisp in some orchards in the Mid-Hudson Valley region, and organic production in the southern part of the state that went up to 100%. But if we would average all these, I think they would be around 20%. 20 to 25% would be the average that we have seen across the state. Certain years we did not see outbreaks, but the fungi are present. We have seen some orchards that have not been sprayed, so it's there. It's just, maybe the people are spraying really well. And we have seen outbreaks in 2018. So why do we have more issues with bitter rot now? If you ask Dave Rosenberger, who has been in my position in the last 30 some years, the most important trick that led to more issues with bitter rot is the implementation of 77 days of pre-harvest interval for mancozeb, because mancozeb is very effective against bitter rot. We do tend to have in the last, I would say 5 to 7 years, more hot and humid summers with very warm wetting events that come with it, which is the weather that really favors these pathogens to infect. We do grow more highly susceptible cultivars, and then sometimes there is a chance that this pathogen really thrives well because people are not removing fire blight strikes from the orchard as diligently as they should, because the fungus inhabits the dead tissue of the fire blight strikes, and that's where it also can overwinter. And the orchard sanitation, if it's not applied to remove the decayed fruit from the orchard floor and mummies in the canopy, it seems that really contributes to this pathogen to survive better. So if you have a grass strip below your trees, it seems that that's actually good because the grass strip allows the fruit actually to decay better because the activity of different flora and fauna there is much higher, so the fruit degrades faster than the actual herbicide strip. They just stay there mummified. So the late maturing cultivars might also have issues with not enough residue, fungicide residue late in the season, and that seems to contribute allowing this pathogen to infect. And then we also might be having issues with people implementing or activating irrigation on time. Usually it's the time when people actually see the trees being water or drought stressed that they turn on the irrigation. And that's too late because the drought-stressed trees have compromised immunity, especially the fruit. And that seems to contribute to the infections on the fruit because the immunity of the fruit itself in the skin is compromised, and that allows not only different physiological issues to happen, but also for fungi to be reactivated and infect. So we have many open questions about bitter rot. Do we have this dormant infection? So sometime, during the petal fall, there's definitely reports in different parts of the world that these fungi infect during the time. They can infect as early as petal fall or bloom, but they stay dormant or quiescent up until the time there's certain cues that they reactivate them. So we don't know whether that happens in New York, but it's highly likely. We do not know, and there's no research whether heat fruit injury contributes to these fungi, to infection. And sometimes, we can't correlate, or sometimes you can correlate also sunny side of the fruit that is very exposed to the sun and stressed to the incidence of these diseases. And also, we just don't know which species are present in New York, and the species can vary widely in their biology, but also sensitivity to fungicides. And different farms have different management practices, so that can also contribute to the different incidence, depending on how you approach your management. So definitely, there's many different things that can be researched here, but what we wanted to do is we just wanted to see what we have in New York, and this is just a small overview of these two groups that you see here. The blue one at the top, called Colletotrichum complex. This is the group of species, okay, from the same genus Colletotrichum. So it's a complex of species, meaning there's multiple species within it that cause bitter rot. And the ones in yellow that I highlighted here, that are in the blue group, which are more accustomed and more prevalent in cooler regions, those yellow ones are the ones that are so far reported that cause bitter rot in different parts of the eastern board and states. And we have not found these ones in the white. But the Colletotrichum diosporoides group, the red one, that is the second in line, is more present in warmer regions. And the first four also have been described on pome fruit, but we didn't know up until just this past beginning of this July which species we have in New York. And that's what my research lab investigated. I'm going to show you that in a minute. So, but before I go in and explain that, I just want to give you the brief justification of why do we need to know the causal species. Well, research has shown that if you look at the Colletotrichum complex of species, that first one that is more accustomed to cooler regions, we do see more tolerance to fungicides in that group versus the other group that is more present in the warmer regions of the world. For instance, the orthopsin and raleigh might be definitely not working as well against the Echinidum complex species, such that's your Colletotrichum fiorini as a species, whereas these other ones will be more effective against the other group. So we don't know too much about the tolerance to these fungicides between different species. We don't know which species we have in New York. We also don't know what the susceptibility is to the DMI fungicides, such as raleigh, one of them, and also there is whether we have resistance to the QY fungicides, the FRAC 11 group, which is your Pristine, Marivon, and Flint, for example. And then the new class of fungicide SDHIs, such as Frontelis and Marivon, and these different fungicides, we just have preliminary data from different other parts of the world that they are hit or miss. Some of them are effective, some of them are not effective in certain species. So it really depends. And then if you go in different parts of the world, you have variation between those parts of the world in which species you have. So Just if we go to Pennsylvania, you have a different complex. If you go to Kentucky, you have a slightly different, slightly more different complex than us. One thing that we do see is the Colletotrichum equitatum complex species. They grow to the, at the temperatures below 41 Fahrenheit, whereas that's not the case with the species from the Gliosporides complex. So they definitely, based on research, differ in their susceptibility to fungicides, and we just don't know how they differ in the other factors, such as ecology, what are their temperature requirements, in their virulence as well, and that's like an open field that we wanted to go and investigate. We sampled these different farms that had issues with bitter rot, and with 2 of them on Long Island and most of them in the lower Hudson Valley. We sampled roughly 16 farms in the Hudson Valley, and we have 5 or 6 more farms that are outside of the state in Virginia and Pennsylvania. And we collected around 400 isolates, and then when we did the isolation, we have conducted a deep sequencing analysis that led us to identify that 60% of our collection that we have collected from these farms, 60% of them were the bitter rot caused by Colletotrichum fiorinea. That's the species in white, font up there. 25% of them were caused by Colletotrichum chrysophyllum, the species that has not before been reported on apple. So we are a little bit dumbfounded with that because we did not expect that species. It's known only on banana so far and on cashews, okay, in the really tropical and subtropical parts of the world. And we have found it here on apples. And so that's the species from that group that I mentioned to you is more prevalent in the warmer regions of the world. And then 12.5% of the samples consistently throughout the 2 years, we have seen this new species that we named in our research recently as Colletotrichum novaburracensi. The novaburracensi is the name of the species that is Latin translation of New York State. That's the way how we wanted to describe it. It's a pathogen within that group of more warmer prevalent species. It falls in that group, and we have found that one. That was the big surprise. We did not expect to see at least 2 new species for this host, and 1 new that is not as described before. We expected some other species like Fructicola or CMNC and other ones that are described in different other states like Kentucky and Pennsylvania, but lo and behold, that was not the case. So this just came out just several days ago. So in a journal, we just we published this description of the species, which is something that really contributes to other researchers a lot. There's a lot of people in different parts of the world working on these, not only on apple, but on mango, on all these other crops, field crops, fruit crops, name it, they have it. And also the nursery crops as well, flowers and stuff like that. So it's a really a huge field of research. And so we're happy that we were able to report this because we believe this is going to contribute to the better way of management in the future. So talking about the varieties, this is a slide I mentioned briefly the varieties that they contribute. If you grow more susceptible varieties, you might be having more issues with bitter rot. So this is what my colleague from University of Georgia has in his presentation, Philip Brannen, who actually just summarized briefly this study from Alan Biggs that showed that if we look at this, they looked at 1 species called Colletotrichum equitatum from that first group that likes more cooler regions. You can see the Honeycrisp and Pristine in their research was very highly susceptible, or the most susceptible, including ginger gold, whereas the least susceptible would be your Fuji, or moderately would be maybe Golden Delicious and Gold Rush. So we did not have any data for New York in terms of this susceptibility, and we did not even know whether we should expect to see any differences. But this is what they found, in Virginia and other parts of the eastern board. And what we did is we took 2 species. So the one that we tested is the one called Colletotrichum fiorinea that is the most dominant that I mentioned to you. And then we tested Colletotrichum chrysophyllum. And it seems that there's a little bit more virulence or aggressiveness of the chrysophyllum in terms of the size of the lesion that it causes on these different fruits that we inoculated. We infected them with these fungi. So it seems that there is a little bit more virulence in these species that are more prevalent in the warmer regions in comparison to the fiorinea. it seems that, overall, if you would average these across different cultivars, that they have a little bit less virulence in them. So they can vary in their, ability to cause a disease. Also, what we did, we tested this at another stage. So the first image I showed you was when the fruit was about an inch size in diameter, and this is the near maturity. So these fruit all were picked up around the first week of September when Honeycrisp harvest time is. And we also can see the similar trend that overall that these species like Chrysophyllum, which we chose here as a representative, is slightly more aggressive in comparison to the Fiorinia. It has, larger size of the lesions that it causes, that to us indicates that they differ in their biology and in ability to cause a disease. So we also went on and then tried to compare all three different species. So these three bar colors that you see, the white, the gray, and the black, are three different species. And within each cluster of these three colors, you see different cultivars. So ranging from Fuji, Gala, and Red Delicious, and Golden Delicious, and Honeycrisp. And you can see here that we have statistical differences in their ability to cause a lesion or the rot on fruit, okay? And, specifically showing here that maybe on Gala, the Chrysophyllum and the Novaburracensi, the species that are more accustomed to warmer regions, are more virulent on Gala than maybe Fiorinia. And you can see that each of these different cultivars have slightly different responses, and they are statistically significant. So we, reasonably believe that they have different compatibility with different varieties because they have different pathogenicity related characteristics. And so it's important to know the species that you have in your orchard, because then the management can be done better based on this. In terms of the life cycle of this pathogen, so this is a simplified life cycle. If you start from the bottom of this graph here, or maybe on like a cartoon that I just pasted there, you'll see that the pathogen overwinters in the buds and mummified fruit and in the cankers on the wood, and also on the brush that is left over on the ground, but also under the bud scales of the alive buds. And in the spring, the rain allows the pathogen to splash its spores from these mummified fruit and cankers and buds and onto the flowers, and eventually on the fruit during the summer. And then the pathogen forms new generation of asexual spores. And all these organs that are infected that you see in spring and summer can drop down on the ground and allow the pathogen to bridge over the time up until the next season. And eventually the fruit also drop, the flowers and the leaves can drop and also the brush that you leave there that is left on the ground on the orchard floor can allow this pathogen to overwinter and carry over to the next season. So in general, the survival is definitely multi-pronged. There's multiple sources of inoculum. There's also a very good evidence that's going to come out soon in a research paper that this pathogen survives very well on many different forest trees as an endophyte. So meaning it just dwells there in the leaves, does not cause any symptoms, and the tree has it, and then eventually just spills over into the orchard. The spores the pathogen releases in the spring and summer, they are rain splashed, and they can be dispersed throughout the whole season. There's a huge infection pressure, meaning there's a lot of spores to be released both from the forest and also from the inoculum sources within the orchard, and that continuously is happening through the season. Sometimes as little as 5 hours of leaf wetting is enough for infection to occur. So basically those long warm wetting events increase the infection severity. And when you see it, that means that the fungicide residue was not there, but also that the weather was very conducive. The fruit usually get infected. They can get infected as early as bloom, but we don't know whether that happens in our conditions yet, and people are still investigating that on the eastern board. But majority of infections usually happen mid to late season. So basically right about now up until the end of the season. That's when people start, noticing it. And we see these severe epidemics early in the season when we actually have conditions right about like we have right now in the lower Hudson Valley, very warm weather that is followed by rains. So that weather is allowing the primary infections to allow, to happen, and then those infected fruit form new spores that then they carry out and they actually conduct secondary infections, and then we have a big headache because of that. So this is another just cycle that I just wanted to show you. This is the way how the reproductive structures of this fungus look like. The bigger circle that you see all around shows the sexual stage of the fungus. So some species are known and reported to have sexual reproduction. So on the outer left side, you see perithecium there, the fruit, the pear fruit looking like a fruiting body that forms ascospores and they can infect. And if you go to North Carolina, you'll find from Sarah Villani that she is actually monitoring these spores to detect when they are actually released to allow infection. But you know, I don't know whether we have them in New York. But the inner circle that you see on the right side is the reproduction with asexual spores or conidia. And they can, you know, the bitter rot fungi can have multiple generations of conidia. And one thing with this arrow that says quiescent latent period, I just want to emphasize to you that this pathogen as well seems to be dormant after the infections during petal fall, and it can dwell on the skin that you cannot see the symptom that whole time up until the point when fruit starts to mature or certain other stress factors play into the fruit immunity system, dropping the fruit immunity system, and then the pathogen reactivates and infects, and you can see the symptoms. So that's the problem also with this fungus as well, that most likely it has that quiescent latent period as well as the black rot fungus. So what we wanted to go and try to do is see if we can actually isolate this fungus in the middle of January and see if we can, how many buds can actually contain this fungus. So this is a summary from 2 years of data. And these are the varieties that we tested that you can see on the left side in the first column. If we would summarize all that, we found that out of 100% of the buds that we collected over 2 years, we found this pathogen, these pathogens overwinter at 15% of the buds. So if you had infection last year, there's a high percentage of chance that inoculum is going to be surviving really well under the bud scales of your regular fruit buds that are going to open next year. And out of those 15%, we had 10% of them where the species from the Gliosporides complex were. Like specifically, I'm telling you about those 2 new species that we detected, so the ones that prefer warmer regions. And then the 4%, so the smaller percentage of them, were the ones containing species that is more prevalent, the Fiorinia species that is more prevalent to the cooler regions of the world. So that's kind of like interesting, and we did not expect that. So definitely it seems that in New York, the buds can be a source of inoculum, but we need to repeat this a little bit more, and with more sampling. We just had 2 locations here, so we want to do more with this. In terms of the fungicides you can use, these are the main groups, or the FRAC groups that I would mention to you here. The FRAC is an acronym that stands for Fungicide Resistance Action Committee. There's a group of scientists that work 24/7 to prevent that the resistance to fungicides occurs. And basically, they actually recommend these codes that you see here, such as code 11 for QI fungicides. The MBC thiophanate methyl belongs to the thiophanates, which is your MBC group 1, DMIs, FRAC3, and SDHIs. And I mentioned here omega-fluazinam, which is a new group of the fungicides, and we don't know whether it's effective against bitter rot. But if you look at these first 5, those are actually the fungicides that you are dependent on to control bitter rot, especially on the QIs. So we test these fungicides in the lab. So this graph shows you 3 different species: the Fiorinia that loves more cooler regions, and the black and the gray one that love more warmer regions. You can see there is a huge variability how they respond to different fungicides. So for instance, difenoconazole, which is the far left one, seems to be quite effective, but there is still difference in different species how they react to different fungicides. And if you go to benzovindiflupyr, which is your 4th from the left, you can see here that in this case, there is more tolerance of the Fiorinia species to that fungicide than maybe the Novaboracensia or Chrysophyllum have. So definitely, this is an important factor in the way if you want to manage bitter rot in general. Which fungicides you combine in tank mix and how do you alternate them seems to be very important. So in the orchard, in the way how I would advise people to approach bitter rot control is that you should always have an assumption in your mind that apple fruit are susceptible at all growth stages of growth, but definitely more when they start to mature. that if you have weather like we have right now this July, when we have very warm and hot days that really precede warm weather events, that those are going to be the prime infection events that are going to allow infection by these fungi. Never delay your rot sprays beyond the 10th July. But keep in mind that infections can start as early as 1 to 2 weeks after petal fall, and they can the infection period, meaning the spores can be produced by the fungi all the way up until the harvest. So it's a completely contiguous block of time when you can have infection. If you do spray, try to align, your QOI sprays, so your Pristine, your Flint, your Sovran, or your Maravon. You should try to align them before the warm weather events, because those are the most severe ones that you should protect against. And if you can, you should keep up with the rain. If the rain is frequent, like it is in these past several weeks, you should definitely spray on an interval 10 to 14 days. When you get to more drier parts of the summer, you can stretch to 21 days. But if you get 2.2 inches of rain, it doesn't matter when you sprayed. You have to go out and repeat that spray that you did, even if it was 1 or 2 days before, because you lose that residue on the fruit that really protects the fruit. So that's what you need to maintain on the fruit. Some late maturing cultivars, like that mature up to the beginning of November, they have to have continued residue protection with fungicides up until the harvest. In general, what I wanted to say is that even though that we are dependent on these QI fungicides like Flint, Pristine, and Maravon, and Sovran, and Luna Sensation, you can definitely always tank mix with Xyrem and Furban and Captan, and you should, for anti-resistance management strategy that basically prevents potential development of resistance to QOIs in these fungi, which we don't want to have. And Topsin, you should bear in mind, and the DMIs do not seem to be very effective against the bitter rot fungi, which is kind of something that really bothers me right now, because if you look at the data from Georgia, which I'm going to show you right now, it seems that it's very effective. Inspire is very effective against bitter rot. So we don't have the data. We're going to try to get the data. So if you use Captan alone, besides mixing it with different fungicides, you should go with higher rates, 3 to 4 pounds, or between 2.5 to up to 5 pounds of Captan of the 80 WDG formulation, because you can't rely only on a low rate when you have high infection pressure. You can, even with a high rate, you can lose the battle with bitter rot, under the high infection pressure. So you have to increase that rate if you're using it alone. So basically, the overall idea would be to combine any of the QI fungicides, the FRAC 11 group, with any of these contact fungicides such as Captan or Xyrem or Furban. And they're going to be, Captan and Xyrem is going to be effective against Sudwatch and fly stick as well. They're going to help you against it, but primarily the biggest muscles are going to be by the QI fungicides. They are also effective against Sudwatch and fly stick. To prevent any storage incidents of bitter rot, what we would definitely recommend is that you keep up that residue up until the harvest, and that if you can, you cool the fruit after you pick them immediately at 40 Fahrenheit. That seems to slow down the development of bitter rot. So if you can do that, if you're able with the capacity of cooling, you should do that because it helps a lot. And then in post-harvest mode, you can apply scalar fungicide for dioxanil, but it can only be effective against fresh infections, meaning the infections that just set in before you harvested, not the ones that progressed already. So there is some benefit of scalar there. It seems to be effective. So one thing that is difficult, if we look at this strategy for bitter rot control is, and many people complain to me about this, that we have to limit the number of QI applications to 4 sprays per growing season. And the reason why I am really, really pushing for you guys not to use more than 4 sprays is because we do not wish, for these Colletotrichum species that cause bitter rot, we do not wish them to become resistant to QI fungicides. And knowing the history, and some farms applying 6 to 8 sprays of QI fungicides throughout the summer, that's just calling for trouble in the future. And if you can, you should probably back off from using any of the QI fungicides, Luna Sensation, Flint, Pristine. Not use those early in the season for powdery mildew. Just focus primarily on the DMIs, primarily on Raleigh, Rime, Indar, and Procure, and finish your early season that way, and then save these 4 QOIs for later in the season for bitter rot. That's the goal that we have to achieve if you can. So, for instance, if you had issues with powdery mildew last year and you don't want to have this issue, you should maybe start a stage earlier at tight cluster. Never miss your pink and petal fall spray with DMIs, and then you should be okay with powdery mildew without using the QOIs. And then if you do deliver during the summer, your Flint, Flint Extra, Sovran, the QI fungicides, Pristine, and Lumen Sensation, you should definitely do it just before the hot summer days followed by rain. I know that that's not optimal every time because there's probably more infection, more like rain events that can trigger the infection. But these 4 applications of QIs per season, you can alternate them with these middle group of Captain with Inspire Super, or Captain plus Prophyte, or Captain plus Topsin, or Furbam and Xyram. So these ones, these contact fungicides such as Captain, Xyram, and Furbam should definitely be tank mixed with the QI fungicides when you apply QOIs, but then you can also use them standalone at the high rate, and also you can mix these so that you can bridge over the time up until you come to the harvest. So usually we end up having between 6, 7, 8, or 9 cover sprays, depending on the year, depending on the amount of rain that we get and the amount of rain events. But in general, I would advise you to use Mancozeb early in the season. That is going to aid you controlling the scab and rust. Then switch to the Captain schedule for the cover sprays. If you do opt for using Prophyte, you should definitely be encouraged to use Prophyte because this product does not cause any resistance. It's not prone to causing resistance in these fungi. It actually acts by boosting the immunity system of the fruit and the plant. So it does bear a benefit there, and it's also effective against, it can help against Pseudobacterium fly speck. When you alternate the fungicides, you should definitely keep alternation, because in this manner, you should apply you could apply 2 QIs in a row, then you should not apply the 3rd one in a row. You should definitely do a different combination. And some of these combinations that I mentioned here, you can apply 2 in a row, and to bridge over up until the harvest. When you get the 2-inch rain event, you should definitely respray. So whatever you tank mix, bear in mind that you're broadening the spectrum of efficacy against these fungi, and what broadens the efficacy also helps you prevent the resistance to move into these fungi against the QI fungicides, which is exactly what we want to achieve. So we are actively working to expand this in the list of options for you in the summer. So we have a big project funded by the Farm Viability Institute this year and the next year that is going to help us maybe see which fungicides might be the new ones that we can implement in the future. And just to show you that the situation is not as dim in the New York as it is in the southern part of the Eastern Board, this is just a list of the treatments that Philip Brannen from University of Georgia has compiled of what is actually legally allowed to use. And I'll focus your attention to the bottom of this slide, where the cover sprays start. So the second cover starts down there with yellow, and then you switch to the white cell there with cover spray 3, 4, 5, 6, and 7. And one thing that really is interesting is, yes, you can see there that they apply Mancozeb with Prophyte, and they also mix it with Top Guard. They have Maravon and Captain and Prophyte alone. And they sometimes do 2 or 3 in a row of these combinations, okay? But what really bothers me is, look at how many cover sprays they have in total. In the southern parts of the United States, they go up to 21 covers during the summer. So talk about intensive production of apples there. And that's the reason why this is the case with them, is because they have a longer season there than we do. So they have these many options that are shocking to me, such as Inspire Super. They cover 9, if you can see here, they recommend Inspire Super. It seems to be effective against the glomerullar leaf spot, which is the leaf form of this disease, of the bitter rot. And then they alternate in also the Omega, the fungicide that is new that I just mentioned to you, which is from FRAC 29 group. And they also alternate Inspire Super. So based on our experience, Inspire Super, it seems not to be effective against bitter rot, but they have some data to prove that it is effective in the southern parts of the states. That might be because they have different complex of species in those states versus us. But we usually end up season, maybe in the worst case scenario, with 9 covers. They can end up season with 21 covers. So that's quite intensive. So what about organic orchards? And I know these questions come up a lot of times. Yeah, that is a problem. Okay, bitter rot is the biggest problem after rust that people are worrying about. Dormant copper is going to help you, but it's just going to reduce the overwinter inoculum. It's not going to go and help you with the seasonal infection pressure. If you do apply sulfur, maybe it can help, but you should use the fine ground sulfur, and you have a limitation that you can apply it up to petal fall, because after petal fall, sulfur, too much sulfur actually can reduce the photosynthesis, and you don't want to have that. You want those fruit to grow from the photosynthesis of the leaves, and it can burn leaves if you apply it to temperatures above 90 Fahrenheit. Cueva and Bacjex2 can be applied at low rates, but you might be having issues with fruit russet. Some growers tell me that's not the issue for them, because if it's organic, they can sell it. And then the dependence on liquid lime sulfur, you could mix liquid and maybe control a little bit bitter rot with liquid lime sulfur in combination with low copper rates. However, the liquid lime sulfur has a big issue with it. Usually people use it for sooty blotch and fly speck control, but you have to use it so frequently that in some cases, the oil that you add to liquid lime sulfur can bring this mix to cause injury on the fruit. And that might completely increase the amount of rots that you can have there. So that's the issue with organic production. The list of materials you can use is quite narrow. If you do have an organic orchard, the cultural practices are your best friend. The position where you plant your orchard has to be such that you have perfect aeration or air circulation through the orchard. Definitely have great sanitation to remove anything that might carry the inoculum there. When you do tree pruning, make sure that you do it perfectly so that you have good light penetration and to allow fast drying of the leaves after the rain. And you should avoid planting on poor sites that are low. And if you do have irrigation, that's definitely something that you have to have in these orchards so that you can prevent the drought stress in the trees, which kind of seems to exacerbate bitter rot. There are differences in the cultivar susceptibility, and I just listed you here some varieties, okay? But depending on which pathologist you ask, they're going to go and say that there is no resistance in these commercial varieties that we depend on to sell the fresh fruit. There is some germplasm out there that has been discovered that bears the genes for resistance, but it has not been bred into the commercial cultivars yet. If you do harvest the fruit, always try to cool them as much as, as quickly as possible below 42 Fahrenheit, because that's going to slow down the growth of these fungi, specifically of that group from the warmer region that I mentioned to you. Some tests have been done with Cueva and double nickel, and I'm going to show you these graphs. So this is the Keith Yoder's work that has been done at Virginia Tech that shows that if you apply overall the OSO, the new fungicide, which is polyoxin D active ingredient, it seems that to control this, to control overall the rots, and if you look at the right graph, it seems to control with 9 cover sprays from petal fall till the 9 cover. If you apply this, product, it might control bitter rot pretty well. However, I'm not sure which species they have in Virginia, and also whether this also fungicide would be effective against the species we have in New York. The reason why I'm putting this slide here, okay, which shows the white rot on the left graph and the alternaria rot on the right graph, is to show you just that something that works against bitter rot might not be effective against white rot. So it's highly specific. Like in this specific case, the new fungicide that you see as a green bar on the left might not be effective against white rot at all. Whereas for alternaria rot and bitter rot, it might be very effective if you apply it as frequently enough. So these new chemistries that are coming out might be completely hit or miss depending on which rot you might be having issues with. And finally, this is work from Jeff Alejandro in Western New York, which shows that if you have a spray program, which is the blue bar that you see there flattened as a pancake, that's your Cueva and double nickel mixed in with alternating spray program with also this new fungicide called polyoxin D. And it seems to be working quite well in this year. So it was very comparable to the grower standard. So hopefully in the future, these get labeled or maybe used more by the organic growers.